For the video today on white blood cells, you should be picking up as many of the colors of blue, red, purple, pink as you can. Red and a blue would be fine as well. The first cell that we're going to draw is not a cell at all. It's important to state that right off the bat. This is a red blood cell or an erythrocyte. It does not have a nucleus, so it is not actually a cell. What we want to do is we want to somehow recognize that this has sort of like an outer rim that's thicker and then an inner part that's thinner. It's sort of like a donut that's been filled in the middle, but only with like a translucent structure in there. So I'm not sure exactly how to portray that, but I go ahead and draw it just a darker red on the outside and then a lighter red or pink in the middle. So this is a red blood cell, RBC, erythrocyte. Any of these names are fine. Next, we're going to be starting to draw our white blood cells, and I'm going to push forward the idea of size here. In almost every case, the white blood cells will be larger than the red blood cells, so be careful. I'm going to be kind of drawing more unstructured, not quite such a perfect circle, because that's kind of how they are. This first white blood cell we're going to be drawing is a neutrophil, and I'll label it in a moment. The things that stick out about the neutrophil is that there are these little dots inside of there, granules. And then there is a lobed nucleus that usually has kind of like 3 to 4 bulges. So it's this weird looking nucleus, very recognizable once you see it enough times. The next one we're going to draw is going to be an eosinophil. Now this also has granules, so you should put some granules in there. I am drawing it in pink, which isn't the best color to use, but there is that typical picture of an eosinophil. Now the nucleus of an eosinophil looks a lot like a horseshoe. The lobes here are real thick, and it does tend to be only 2 of them. They are hooked together just as you see. Now you might be thinking, well, that neutrophil at the top and the eosinophil underneath of it look an awful lot the same as each other, and they can be mistaken for each other, but not usually. It's not really something that, like, once you see enough of them, you'll be able to tell the difference. So take a look at, watch out for that, neutrophils versus eosinophils. The next cell we're going to be drawing is a basophil. I'm going to draw it lighter than it usually comes out. A basophil really looks just kind of like this very pigmented structure. There are granules inside of there. There's sort of this usually 2-lobed nucleus that's kind of tied together at the middle. But it actually comes out in practical terms, it comes out much, much darker than this. And it just kind of looks like one big blob in there. So all 3 of these are considered to be granulocytes. At the top is the neutrophil. In the middle is the eosinophil. And at the bottom is the basophil. And they are called granulocytes because they have granules inside of them, which each have their own kind of purpose. And you'll notice that all 3 of them are substantially larger than the red blood cell. Like, you should see just as, that tick up of size larger than the red blood cell pretty much every time with these 3 cells. The next cell we're going to draw is a lymphocyte. There's 2 types of lymphocytes, B lymphocytes and T lymphocytes, and actually there's many subtypes of these as well, but we're just going to draw one. Not being histologists, we don't really need to know, we don't need to parse the difference. You'll notice that this is slightly smaller than the granulocytes, but slightly bigger than the red blood cell. Typically, the red blood cells are the smallest structure, and then the lymphocytes that we're drawing here are going to be a little bit larger than that. Sometimes they can get to be much larger, but typically we see them just like one bit of size larger than the red blood cells. Now the next structure we're going to draw, I'm drawing it substantially larger. This is called the monocyte, and the nucleus is sort of as shown. But what really sticks out about the monocyte is that it is just substantially larger than anything else. with both the lymphocyte and the monocyte, there are not granules. They do tend to be they have a pigmentation to their cytoplasm, but these are not granules. They're not the punctate polka dots like we drew with the granulocytes. So as we label these then, these are agranulocytes, the letter A meaning without granules. These are agranulocytes. We'll go ahead and draw the line between them so you understand the top 3 are granulocytes, the bottom 2 are agranulocytes, no granules. The top one is the lymphocyte, and the bottom one is the monocyte. Let's go ahead and draw the size of all these, and I'm going to be using this U. This is actually a mu symbol that means micro. The red blood cell, its diameter tends to be about 7 to 8 micrometers, that mu M. Neutrophils 10 to 12 micrometers, eosinophils 10 to 14, basophils 10 to 14. So again, you see that that level up from the size of a red blood cell. Now you'll notice lymphocytes have a much wider range, 5 to 17 micrometers. I usually see them probably at about that, I would say at about a 9. So just a little bit larger than the red blood cells in comparison. And the monocyte is one of our larger cells. The monocyte can be as large as 24 micrometers and often is. But even the smallest size you'll notice is 14. And that is just larger than anything else that you see in this picture. Beneath the line there are all 5 types, like just these broad categories. There's many details underneath it there, but the broad categories of white blood cells is that there's 5 different types. 3 granulocytes and 2 agranulocytes. At the top we have our red blood cell, and then we're going to go ahead and draw one more formed element. Again, this is not really a cell. Just as the red blood cell is not a cell because it doesn't have a nucleus or organelles, the platelets also don't have nuclei or organelles, so they're not actually cells, but they're definitely things that you should be able to identify. You're probably going to want like a dark purple and a light purple, or maybe a blue and a purple or something like that, just to be able to differentiate a little bit. Now, platelets are quite small, and you'll see them sort of as dots, and I'm going to go ahead and draw them with little granules inside of them, which they do have. But in a blood smear, you're probably not going to be able to see the little dots. You're simply going to see these little specks of pink usually. Those little dots are not artifacts of the fixation process. They actually are platelets. So again, at the top we have red blood cells and platelets, which are formed elements and not actually cells. The platelets tend to be 2 to 4 micrometers in large. So again, at the top we have our red blood cells and our platelets. You see the size there. The red blood cells and platelets are called formed elements and not cells because they do not have a nucleus, they do not have organelles. And below that we have our 5 actual cells that we find in the blood. All of the white blood cells, or the broad categories of white blood cells, are listed there.